एवरीवन, वेलकम बैक टू माय यूट्यूब चैनल। आज की वीडियो में हम बात कर रहे हैं टी लिम्फोसाइट्स की। थाइमस ड्राइव टी लिम्फोसाइट डेवलप इनटू द इफेक्टेड सेल्स ऑफ द सेलुलर इम्यूनिटी एंड हेल्प बी सेल्स टू प्रोड्यूस एंटीबॉडीज अगेंस्ट द प्रोटीन एंटीजन। T cell constitute 60% to 70% of the lymphocyte in the peripheral blood and are major lymphocyte population in the splenic, periarticular sheaths and lymph nodes and in interfollicular zones. T cell cannot recognize free or circulating antigen. Instead, a vast majority, more than 95% of the T cells, sense only peptide fragments of protein displayed by the molecules of the major histomatically complex MHC. Discuss in more detail. Because T cell antigen receptor have evolved to see MHC bound peptides on the cell surface, T cell only recognize the antigen presented by other cells. The outcome of this interaction varies dramatically depending on the type of the T cells that is involved and is identically to the other interacting cells, ranging from killing of the virus infected cells to the activation of the phagocytes or B lymphocytes that have ingested protein antigens. Peptide antigen present by the MHC molecule are recognized by the T cell receptors. A heterodimer that in most T cell is composed of the disulfide linked alpha and beta protein chain. Each chain has a variable region that participate in a binding a particular peptide antigen and a constant region that interact with the associated signaling molecule. TCRs are non-conventionally linked to a cluster of 5 invariant polypeptide chain. Their gamma and alpha E proteins of the CD3 molecule complex and 2 gamma chains. The CD3 protein and gamma chains do not bind antigen. Instead, they are attached to the TCRs and initiate intracellular biochemical signals after TCR recognition of the antigen. T cells also express a number of the other molecules that serve important function in immune response. CD4 and CD8 are expressed on the distant T cell subset and act as a co-receptor during T cell activation. During antigen recognition, CD4 molecule on T cells bind to class 2 MHC molecules. CD4 expressed on the 50 to 60% of the T cell, whereas CD8 is expressed on about 40% of the T cell. CD4 minus and CD8 expressing T cells called CD4 and CD8 plus cells respectively perform differentiate but overlapping functions. CD4 T plus cells are called helper T cell because they secrete the soluble molecules, cytokines and B cells to produce the antibodies and also help macrophages to destroy the phagocytes microbes. The central role of CD4 helper cells in the immunity is highlighted by the severe compromise that results from the destruction of these cells by human immunodeficiency virus infections. CD8 plus cells have also secrete the cytokines, but their most important role is to quickly kill the virus infected cell and tumor cell. Hence, they are called cytotoxic T lymphocytes. Other important irrelevant protein on the T cells include the CD28, which function as the receptor for the molecule called moleculars, and they are induced on the APCs by the microbes, and various adhesion molecule that strengthen the bond between the T cells and APCs and control the migration of the T cells to the different tissue. T cells that function to suppress the immune response are called regulatory T lymphocytes. These T cells is described later in the context of tolerance of the self-antigen. While most T cells express TC are composed of alpha and beta chains, as a minority of peripheral blood T cells and many T cells associated with the mucosal surface and express TCR also are composed of gamma and gamma chains. They are similar but not identical to alpha and beta chains. Do not express CD4 and CD8, recognize the non-protein molecules, but their function roles are not well understood. Another small population of the T cell express the markers of the both T cells and NK cells. These are so-called NK T cells recognize the microbial glycolipids and may play a role in defense against some infections. यहाँ तक कोई भी बात समझ ना आई हो तो पूछ लीजिएगा। इजाज़त चाहूँगी। अल्लाह हाफिज़।